Imagine you have this really aggressive cancer, and right now the ultimate cure might actually exist.
Right. But getting it it's a total logistical nightmare.
Exactly. It's an absolute nightmare. You have to get your blood extracted, ship it across the country to some specialized facility, and then you spend hundreds of thousands of dollars just to rent space in a sterile lab.
And you're waiting, what, nearly a month?
Yeah, a month. Just waiting for that blood to be genetically engineered and then shipped back.
Yeah.
And by the time the cure actually arrives, assuming it even worked in the lab, it might literally be too late.
Which is just heartbreaking for patients.
It really is. But what if your doctor could just hand you an IV drip and your own bloodstream magically became that $1 million manufacturing plant?
That is the dream right there.
It is. So grab a coffee, pull up a chair, and join us. Because you are the third person in today's conversation. And we're looking at a manufacturing revolution that's happening not in a factory, but directly inside the human body.
It really represents a totally paradigm-shifting moment for how we think about personalized medicine, and more importantly, how we actually scale it.
Yeah, let's get into it. We're diving into a brand new, just groundbreaking study that literally dropped yesterday, March 18th, 2026.
Fresh off the presses.
Hot off the presses. It's titled In Vivo Site-Specific Engineering to Reprogram T Cells. And this was published by this massive team of researchers led out of UCSF and the Gladstone Institutes.
Yeah, it's a heavy hitter of a paper.
It is. And our source stack today is just this one single, dense, but honestly incredibly exciting scientific paper.
Calling it dense is almost an understatement, I think. You're looking at, years of overlapping disciplines, virology, immunology, CRISPR gene editing. It's all finally coming together to solve a very specific physical bottleneck.
Right. And our mission for this deep dive is to sort of cut through all that jargon. We want to understand this massive leap forward and how scientists are figuring out a way to completely bypass that multi-million-dollar laboratory step of CAR T-cell therapy.
Essentially reprogramming your immune system on the fly.
On the fly. Okay, let's unpack this, because to understand why this paper is such a big deal, we have to ground ourselves in the current state of CAR T-cell therapy.
Which, to be fair, is already amazing.
Oh, absolutely.
Wow.
We know KRT is an absolute miracle for certain blood cancers, but right now, it requires what the medical field calls ex vivo manufacturing.
Right, meaning out of the living. You have to physically take the cells out of the patient to work on them.
Right. I always like to think of it as sending your immune system soldiers away to this incredibly expensive off-site boot camp.
That's a great way to picture it.
Yeah, you extract the T cells, ship them to a central facility, and then genetically modify them with a new receptor.
Yeah, the QR.
The KR, exactly, which lets them actually see the cancer. Then you force them to multiply by the millions, pack them up, and ship them back to the hospital to be infused into the patient.
But the clinical limitations of that ex vivo bottleneck are just severe. I mean, the time delay alone is agonizing for patients with rapidly progressing diseases.
They just don't have a month to wait.
Exactly. And the industry, they recognized this flaw years ago. They tried to solve it by developing off-the-shelf or allogeneic CAR T cells.
Oh, right, where they use healthy donor cells instead of the patient's own cells.
Yeah. The idea was you pre-manufacture them in massive batches, freeze them, and then just pull them out of the freezer whenever a patient needs them.
Well, wait, pouring someone else's immune cells into your body? Doesn't that sound like a recipe for a biological civil war?
Oh, it absolutely does.
Doesn't the patient's body just recognize those donor cells as foreign invaders?
It does, immediately. You face enormous hurdles, things like life-threatening tissue rejection or graft versus host disease, where the donor cells actually start attacking the patient's organs.
Yikes. That sounds awful.
It is, and even if you manage that with really heavy immunosuppressants, you ultimately still see frequent relapses.
Because the immune system wakes up.
Exactly. The patient's immune system eventually wakes up and clears those foreign donor cells out before they'd even finish hunting down the cancer.
Okay, so off the shelf is tough. Which brings us to the core hypothesis of this new paper.
Right. What's fascinating here is they're saying, what if we can skip the laboratory and the foreign donors entirely?
Just do it all inside the body.
Yes. If we generate these engineered cells in vivo directly inside the patient's bloodstream, we obviously save all that time and money.
Sure.
But the biological bonus is that we actually create a drastically superior, less differentiated pool of T cells.
Less differentiated.
Right.
Meaning they are younger, like fresher.
Yeah, essentially.
So they haven't been just beaten down by growing in a plastic petri dish for a month.
That is the crucial physiological difference. When T cells are expanded ex vivo in a lab, they are pushed incredibly hard with artificial chemical signals.
Right, to make them multiply.
Exactly. They're forced to divide over and over again. So by the time they go back into the patient, many of them are already approaching a state of cellular exhaustion.
Wow. So they're tired before the fight even begins.
Very tired. Their telomeres are shorter and they're metabolically exhausted. But if you engineer them in vivo, they naturally possess what we call a stem cell memory phenotype.
Stem cell memory. I like the sound of that.
It's huge. Because they matured in their natural environment, they stick around much longer, they replicate more sustainably, and they just fight harder.
Okay, I am totally on board with the idea of in-body manufacturing. It makes perfect sense. But I have a massive structural question here.
Lay it on me.
The human bloodstream is completely chaotic. I mean, it is a rushing river filled with billions of different cells, proteins, enzymes, antibodies, all of it.
It's a very noisy environment.
Right. So how do we actually edit cells safely inside that chaotic environment without, accidentally editing the wrong things?
And that right there is the safety question that has held the entire field back for a decade.
Because if you miss, it's bad.
Really bad. Previous attempts to do this in vivo relied on blunt instruments. They used random integration like retroviruses that just jam the new DNA anywhere they can fit it into the genome.
That sounds dangerous.
It is. It's highly unpredictable, and it can disrupt healthy genes. Or, more recently, they used lipid nanoparticles, LNPs.
Oh, like the delivery system used in the mRNA COVID vaccine.
Precisely like that. But LNPs only provide temporary expression.
They don't stick around.
Right. They drop off the blueprint, the cell builds the receptor for a few days, and then the blueprint just degrades. The cancer-fighting ability simply fades away.
Okay, so you need it to be permanent, and that's where this paper comes in.
Yes. This paper achieves something truly remarkable. They managed site-specific integration of a massive DNA payload directly into a highly specific address in the genome called the TAC locus.
The TAC locus, the T cell receptor alpha constant. So we aren't just throwing the genetic instructions at the cell and hoping they stick somewhere safe.
No, not at all.
We are surgically inserting them into a very specific, predetermined location. I was reading the methodology section, and the way they pull this off, it struck me as being like a highly specialized two-person safe-cracking crew.
A safe cracking crew. Okay, walk me through how you're visualizing that.
All right, bear with me here. So you have this massive DNA payload, which is the new CEOR blueprint, right?
Right.
That's the gold you want to put in the vault. But it's too big to just shove through the wall. So crew member number 1 goes in first.
Okay.
This is what the paper calls an enveloped delivery vehicle, or EDV, and it's carrying CRISPR-Cas9 ribonucleoproteins.
The editing tools.
Exactly. This EDV is basically the blowtorch. It sneaks into the cell, navigates to the DNA, and cuts a highly precise hole right at the TAC locus.
It makes a targeted double-strand break in the DNA? Yes.
Then, sneaking in right behind it, comes crew member number 2.
The payload carrier.
Right. This is an adeno-associated virus, specifically AAV6, and it's carrying the actual CAR DNA donor template. It walks up to the exact hole the blowtorch just made and inserts the gold into the vault.
And then what does the cell do?
The cell panics. It sees the broken DNA and uses its own natural repair mechanism to seal the new gold permanently into the wall.
That natural repair mechanism is called homology-directed repair. And honestly, your analogy perfectly captures the elegance of this dual vector system.
Thank you. I was pretty proud of that one.
It works. The EDV provides the transient temporary cutting tool, and the AAV provides the permanent genetic payload. But, and this is a big but, taking this safe-cracking crew into the real world highlights the exact risk that makes in vivo editing so terrifying.
The off-target effects.
Exactly. If we are injecting gene-editing blowtorches and DNA payloads directly into a patient's blood, what happens if they crack the wrong safe?
Yeah, I was wondering about that. Like, couldn't we accidentally edit a stem cell? Or even worse, what if we accidentally give the cancer cells themselves the CRR blueprint?
The consequences of that would be catastrophic. If you edit a hematopoietic stem cell, those are the mother cells living in your bone marrow that create every drop of your blood. You could cause dangerous, permanent system-wide mutations.
That's terrifying.
And if you accidentally edit a tumor cell, let's say you're treating a B-cell leukemia, and the virus accidentally drops the terror into the leukemia cell itself.
What happens then?
The tumor cell then expresses the CRR on its own surface, It basically binds to itself and masks the very antigen the therapy is trying to target.
Oh, wow. So it puts on a disguise so the real immune cells can't even see it.
Exactly. We call it an antigen-negative relapse. The cancer just hides in plain sight, completely immune to the therapy, and it grows out of control.
Okay, that sets the stakes perfectly. To do this safely inside a living body, this two-person crew needs to be absolutely flawless at picking their targets.
Flawless.
They can't just be good, they have to be perfect.
Yeah.
Which brings us to the technological upgrades. And here's where it gets really interesting.
Oh, yeah.
Because the researchers couldn't just use off-the-shelf viruses for this, right?
They absolutely could not. If you inject standard AAV6 into human serum, the liquid part of our blood, the body's natural neutralizing antibodies will immediately recognize it as a threat.
Because it's a virus.
Right. They swarm it and destroy it before it ever reaches a T cell.
Because, I mean, we've all been exposed to adeno-associated viruses in our daily lives, so our bodies are pre-built to clear them out.
Exactly. We already have the defenses ready.
So how do you get the payload past the immune system's security guards?
You force the virus through an accelerated evolutionary boot camp.
Directed evolution.
Yes. The researchers ran an evolution screen. They took a vast library of mutated AAV6 variants. We're talking millions of slightly different versions of the virus.
Wow, millions.
Millions. And they essentially forced them to run a biological gauntlet. They exposed the virus library to human serum over and over for 3 grueling cycles.
Survival of the fittest.
Pure survival of the fittest.
Right.
They only selected the rare viruses that somehow survived the antibodies and successfully infected human T cells.
And they actually found a winner. The paper details this specific variant that developed a really unique amino acid motif on its surface.
Right, the HAPRVE motif.
Yeah, HAPRVE. And that specific mutation allowed the virus to slip right past the human antibodies. It was enriched, what, 20,000-fold compared to the original library?
2,000-fold, yeah. They dubbed this new supervirus AAV-T7.
AAVHT7. But creating, it wasn't just about surviving the bloodstream, right? It was also about hyper-targeting the correct cells.
Exactly, because surviving isn't enough if you break into the wrong safe. So the researchers ran a massive genome-wide CRISPR knockout screen to figure out the exact mechanical reason how this new AAVHT7 was actually getting inside the T cell.
Oh, I wanted to bring this up because the mechanism here is wild.
It really is.
They discovered that this evolved virus specifically relies on the CD7 receptor. And CD7 is a protein that's heavily expressed on the surface of T cells and natural killer cells.
Right.
So it basically acts as the exclusive docking port for AAVHT7.
Which is a massive safety victory. Because it relies on CD7, it naturally limits the virus from entering cells that lack that receptor.
Like those stem cells we were worried about.
Exactly. It completely ignores those dangerous hematopoietic stem cells we talked about earlier. But remember, the AAV is only the payload carrier. It's only half the crew.
Oh, right. The blowtorch.
They also had to upgrade the blowtorch, the enveloped delivery vehicle carrying the CRISPR tools.
Because otherwise, the blowtorch is just floating around blindly cutting things.
Right?
So how do they tell the EDV exactly which door to knock on?
They heavily modified the surface glycoproteins of the EDV. They completely ablated its natural ability to bind to random cells, and instead they attached a highly specific targeting molecule to its outer shell.
Okay, what kind of molecule?
An anti-CD3 single-chain variable fragment.
Anti-CD3 SCFE. So it acts like a homing missile for the CD3 receptor, which, if I remember correctly, is an explicit marker only found on T cells.
Yes, you've got it. But the genius of this design is that the anti-CD3 molecule does something even more crucial than just targeting.
What's that?
It actually activates the T cell.
Wait, explain that part to me. Why does the T cell need to be activated? I thought we just needed to get inside.
Well, a resting T cell just floating in your blood isn't actively dividing. And if it's not dividing, it won't properly integrate new DNA.
Oh, I see.
That homology-directed repair process, the cellular mechanic that patches the new CAR DNA into the cut site, it only works efficiently when the cell is actively cycling and dividing.
So in a traditional lab, how do they do that?
In a lab, scientists use these tiny magnetic beads to artificially stimulate the T cells and wake them up.
But inside the body, you can't just inject magnetic beads.
No, you definitely cannot.
So by putting the anti-CD3 molecule directly on the delivery vehicle, the EDV knocks on the door, hands over the CRISPR blowtorch, and simultaneously shouts at the T cell to wake up and start dividing.
Exactly. It is an all-in-one delivery and activation vehicle. It's brilliant engineering.
It really is. But obviously theory is one thing. They had to prove this dual system actually worked together and didn't edit the wrong things in a mixed environment.
Right. You have to prove it in the soup.
Yeah. They ran this precision test looking at a common housekeeping gene called CLTA. They tried to insert a green fluorescent protein into it.
Basically trying to make the cells physically glow green if they got edited.
Right. It's the ultimate visual proof of precision. They mixed their AAV and EDV system into a soup containing stem cells, macrophages, and various common B-cell cancer models.
And the result was staggering.
Zero integration in the stem cells, right?
Zero integration in the cancer cells, too. The only cells that lit up green under the microscope were the targeted T cells. Total beautiful precision.
Okay, so we have this highly evolved, ultra-precise delivery system, and they moved to testing it in humanized mice genetically engineered to possess human immune systems.
Right, the next big hurdle.
But I have to ask the critical question here. Is this actually better than the traditional lentiviral methods we already have?
Right.
Because scientists have been trying to do in vivo editing with lentiviruses for a while now.
It is a vital question. And the paper tackles it head-on by benchmarking their new TRC targeted system against the best-in-class in vivo lentiviral approaches.
So how do they compare?
To understand the profound difference in the data, we have to look at how the two systems manage power, or, in biological terms, gene expression.
Oh, okay. Let me try another analogy to visualize this. Let me know if this works.
Let's hear it.
I compare traditional lentiviral editing to plugging heavy-duty space heaters into random wall outlets all over a house. You don't know exactly which circuit you're on. You're just pulling power randomly.
I like where this is going.
Sometimes it works fine, but sometimes you plug 3 heaters into the same circuit and you get erratic, dangerous power surges. That's the random, heterogeneous CAR expression you get with retroviruses.
That is exactly what happens. The retrovirus drops the CAR gene randomly into the genome and uses a constantly on viral promoter to drive expression. It forces the cell to produce the receptor at maximum volume, constantly.
Which has to be exhausting for the cell.
It's incredibly erratic and exhausting.
But this new TRAC integration method, it's like hardwiring a smart thermostat perfectly into the house's main breaker.
Yes.
Because they cut exactly into the TRAC locus. It doesn't use a rogue viral power source. It piggybacks on the T cell's natural promoter.
Exactly. It regulates the new CAR expression perfectly. It turns the cancer-fighting machinery up when it detects a tumor and rests when it's clear.
And that physiological regulation makes all the difference in the world for the longevity of the therapy, right?
All the difference. When the researchers compared the in vivo TRAC-edited cells against in vivo lentivirus-edited cells in a mouse model of BAL leukemia, the data synthesis was striking.
What do the numbers look like?
Well, the lentiviral approach showed highly variable CR expression on the cell surface. But the TRAC-edited cells had completely uniform expression. Every single cell was perfectly dialed in.
And what did that uniform expression actually mean for the cancer fight in the mice? I saw the expansion numbers in the paper, and they are wild.
They really are. By week 2 of the treatment, the in vivo TRC cells showed a massive 21 to 50-fold higher expansion rate in the blood compared to the lentivirus-edited cell.
Wait, 50 times more cancer-fighting cells?
Up to 50 times more, yes.
Why does the smart thermostat effect create such a huge difference in raw numbers?
Because the uniform natural regulation prevents the T cells from exhausting themselves. In the lentiviral models, the cells are essentially screaming at maximum volume all the time.
So they just burn out.
Exactly. They burn out their metabolic reserves and undergo activation-induced cell death. The TRAC cells, however, rest when they need to. The researchers found a drastically higher percentage of those stem cell memory phenotypes we discussed earlier in the TRAC group.
Ah, the durable cells that provide long-term surveillance.
Yes, and because of that stamina, all 6 mice treated with the TRAC in vivo system achieved complete tumor clearance.
All 6.
All 6. Whereas the lentiviral models had spotty partial responses and eventually succumbed to the disease.
Wow. Okay, if we connect this to the bigger picture, solving blood cancers, like leukemia is fantastic, but the researchers didn't stop there.
No, they pushed it further.
Because blood cancers are the traditional proving ground, right? The target cancer cells are freely floating in the same space as the, say, RT cells. They literally bump into each other in the bloodstream.
Exactly. But they expanded the scope significantly. They tested the system on models of multiple myeloma next, programming the delivery vehicles with a CR that targets a specific molecular flag called BCMA.
And how did that go?
Again, they saw complete responses.
That's incredible.
But the true test of this technology's potential was when they went after the final boss of oncology.
Solid tumors,
Yes.
which are notoriously difficult for cell therapies to crack.
Extremely difficult. Unlike a blood cancer, a solid tumor is a physical fortress. It has a dense, fibrotic, extracellular matrix that acts like a literal wall. It has erratic, compressed blood vessels that make it hard for immune cells to even enter the area. And worse, the tumor secretes toxic, immunosuppressive chemicals into its microenvironment.
It's like a castle surrounded by a moat of toxic gas.
That's a great way to put it. And traditional ex vivo CAR-RT cells, the ones grown in a lab for a month, they usually fail here because they are already metabolically exhausted before they even reach the castle walls.
They just don't have the stamina to breach the fortress.
Exactly. But the researchers targeted a highly aggressive type of soft tissue cancer, a subcutaneous sarcoma in these mice.
Okay.
They programmed the dual vector system to build an anti-B7H3 CRT cell. B7H3 is a molecular flag widely expressed across many different solid malignancies.
And incredibly, even against this solid tumor model, their in vivo delivery system worked.
It did.
It generated the fresh, non-exhausted CAR-T cells directly in the body, and those cells successfully trafficked to the tumor, infiltrated that hostile microenvironment, and attacked the sarcoma.
Resulting in complete responses in the majority of the tested mice.
That is just phenomenal.
Because the cells were generated in vivo, they had that stem cell memory stamina, they could continuously proliferate and sustain the siege on the tumor fortress over time.
Right.
This is a monumental proof of concept for the versatility of the AAV and EDV system. It proves this technology isn't just limited to floating leukemias. It has the mechanical potential to tackle the hardest cancers we face.
So what does this all mean? Let's bring this back to you, listening right now. Why should you care about the alphabet soup of AAVs, EDVs, and TreaDC locus integrations?
That's a fair question.
We are looking at a literal paradigm shift in healthcare logistics. For a decade, we've known how to genetically cure certain untreatable cancers, but the logistics made it a boutique, elite, and just painfully slow therapy.
Very slow.
We are talking about taking a complex, bespoke genetic therapy that used to require a month-long, million-dollar hospital ordeal and shifting it to a targeted, off-the-shelf 5E drip that turns your own body into the laboratory.
And that is the core value of this research. Knowledge fundamentally applied to remove a barrier.
Yeah.
We've talked for years about the abstract promise of CRISPR and viral vectors. But this paper takes those raw tools, forces them to evolve past the human immune system, and builds a practical, highly specific delivery mechanism that solves a very real, very painful clinical bottleneck.
It entirely bypasses that fragile ex vivo manufacturing infrastructure.
Completely.
It's staggering to think about. But it leaves me with one final provocative thought.
Yeah.
If we can mass produce these viral delivery vehicles, if personalized genetic cures become as easy to manufacture and administer globally as a standard flu shot.
Oh, the economics of it.
Exactly. What happens to the entire economic model of modern healthcare? Do we just shift the bottleneck from manufacturing cells in a lab to manufacturing highly evolved viral delivery vehicles at a global scale?
It's definitely something the industry is going to have to figure out fast.
Something to mull over. Thank you for pulling up a chair and joining us on this deep dive. Keep questioning the bottlenecks. Keep looking for the elegant solutions, and we'll see you next time.